Ok.
Vai avanti.
Sì.
Condivide le screen?
Sì.
Ok.
Puede ver?
Sì.
Ok.
Ci sentiamo, vado al mio computer. Ciao.
Sentite bene?
Sì.
Ok, so let's start.
Gira la pantalla para que vea los alumnos.
Sí, ahora te está viendo a ti. Ahí.
Ah, ok. Vedo anche. So today I want to talk about my research topic concerning the use of mosses for biomonitoring of air pollution. But firstly, let me explain what I mean for biomonitoring. It is the use of living organisms to understand the level and the type of contamination of a specific environment. When we perform biomonitoring studies, we can apply two types of techniques, bioindication studies or bioaccumulation studies. For bioindication, we use organisms that are sensitive to the pollutants in the environment. So we evaluate the effects of pollutants on the organisms, for example, the morphological changes or the decrease of the number of these organisms in the environment. And we do an indirect evaluation of the environmental pollution. The information that we can obtain is only qualitative because we cannot define the amount of pollutants in the environment, but only the effects of these pollutants on the organisms. When we perform bioaccumulation studies, we use instead tolerant organisms, so very resistant to pollution, but these organisms are able to accumulate substances, contaminants, inside their tissues. So with this manner, if we evaluate the concentration of pollutants inside the tissues, we can understand which are the amount and the type of contaminants in the environment. So this type of studies are direct and quantitative evaluations of the environmental pollution. We can use different type of living organisms, for example, lichens, or also bees, or also, for example, pine needles. We can use plants, so several types of living organisms. But in my case, I used mosses as bioaccumulators. For example, there is a famous network, the UNECE ICP vegetation program. It's a network between several countries in Europe with the aim to evaluate the pollution in the environment, so in specific places in Europe, by analyzing mosses collected in natural sites. But why we use mosses for these studies? Because they have several characteristics, specifically the most important, the high ratio surface of tissues with mass, and especially a great ability to exchange cations on their surfaces. But the most important characteristic is that mosses can accumulate substances, so also contaminants from the environment, without using the roots, but using only the green parts of the tissues. So any type of nutrients and contaminants that can derive from atmosphere can be accumulated by moss tissues. When we use moss for biomonitoring, we can perform two types of studies. One is active biomonitoring, another one is called passive biomonitoring. Specifically, in the latter case, passive biomonitoring, we go in a specific environment. We collect moss, we analyze mosses, and we study the concentration of contaminants in moss tissues. So we can evaluate how much contaminants are present in the place where mosses are collected. But if we use active biomonitoring, we use transplants of mosses. So we take mosses from a clean site, so sites that we consider very, very clean and natural. We take this moss and we transplant, we expose in another place in which we will evaluate the contamination. In a specific case of active biomonitoring, moss bags can be used. For example, my research topic are mostly focused on the use of moss bags. So little envelopes in which we put mosses collected in a natural sites, and we use this system to monitor the pollution in any type of site. For example, in this case, you see an image of a balcony of a house, so inside a city. How we perform this type of biomonitoring? Firstly, we of course we follow several steps. The most important is the selection of moss. So which type of moss we can use for this system and where to take this moss. So we study literature data concerning the use of moss as biomonitors, as bioaccumulators. So if we have a lot of information regarding the use of a specific moss, so we try to collect a particular species. For the collection of moss, of course, we go in places where it is very easy to find the species. And we choose a moss that can produce in the environment a high biomass. So the process of collection and use of moss for biomonitoring will become more easy. After collection, of course, we have to treat the moss because the aim is to use fresh and functional parts of moss, so not the dead and decaying part of mosses. So we took the green apical parts because these parts are the youngest in the moss. After that, we do several washings, for example, using water or EDTA, because we have to clean the moss surfaces. So in this manner, the moss can accumulate the new substances. After that, we do oven devitalization. This treatment is a simple drying in several steps or one unique step, in order to use moss, not living, but dead moss, because we studied that this process, even if we use dead moss, they are always able to catch contaminants. So the effect of metabolism on the capture of contaminants is nullified. After that, after moss treatments, we prepare the moss bags. They are very small. In the traditional style, we use just 0.1 gram of moss for each bag. We try to keep one specific ratio, so 10 milligrams of moss for 1 centimeter of surface area of the bag, because it's important to evaluate how much moss is in contact with the environment. So it's not important only the amount of moss inside the bags, but also the ratio with the surface of the bags. So we simply use a nylon net with the specific diameter of the pores, and practically by hand, we prepare these moss bags. After that, we expose, using the plastic rods, we expose the moss bags in any type of site. For example, in this case, you see agricultural fields, urban environments, so again, a balcony of houses, and another case, an industrial place. This gray building is a monitoring station within an industrial plant near my town, my city. How we expose the moss? Generally, we put the moss bags at a height of 4 meters from the ground. We can use wooden trellis, lamppost, any type of support, depending on the site. Normally, we use 3 replicates in order to have at least 3 data for each measuring point. And the standard period is, say, 6 weeks. Which type of exposure sites? I told you that any type of sites. So we try to distinguish sites with a different contamination degree. Of course, it's important always in this type of study to evaluate also the contaminants in sites, in background sites, because we think that these sites have the lower contamination level to compare with the other type of sites, for example, agricultural environments, industrial sites, in which the pollution level is more high compared to the background. In any type of sites, we can distinguish also sub-sites. For example, inside the urban, inside the cities, we can expose moss bags in the streets, in public gardens, in car parking areas. But also we can evaluate indoor pollution, for example, inside houses, offices, schools, for example. And we can compare the levels of contamination within and outside buildings. After the exposure, we follow with the analytical determination. With only few amount of moss, 0.0 gram of moss, we can analyze any type of contaminants. For example, elements, so metals, rare earths, particulate matter, the fine powder that we can breathe, for example, in the cities or industrial places, but also isotopes, radioisotopes. As the professor told you, I was in Fukushima, so my focus was radioisotope. But also gaseous pollutants, for example, hydrocarbon, polycyclic aromatic hydrocarbons, but also the old substances diluted or present in form of ions inside water. Because basically everything is emitted in the atmosphere and it is removed from the atmosphere in form of wet and dry depositions. Everything arrive on the ground and can arrive on mosses can be analyzed by moss bag biomonitoring. But why we want to use, which is the advantage to use the moss bag biomonitoring? Because with a simple system, we can monitor any type of site. Compared, for example, the traditional monitoring station, and you cannot set monitoring stations everywhere, inside offices, inside the houses. But with MOSBEX, you can monitor any site. You can obtain a high density of measurement points. You can do temporal evaluation. For example, you can evaluate which is the changing in pollution during the season, in summer, in winter, or for example, during years. Another advantage is, as I told you, that with one only bag, you can analyze any type of pollutants. And in general, it's a very easy system to apply and also very cheap if compared with other monitoring systems. Practically, I started my research activity on mosses working within this big European project. So you have the website, you can Google it, and you can find some information concerning. But this project was very important because it was focused on two important aspects. How to standardize the method, because it is only a scientific application. It's not, there is no law, no legislation that can regulate this method. It is only applied within universities or for scientific purposes. So this technique, if we want to apply in the future everywhere, it is necessary to standardize the method. So to give a protocol, a specific protocol for its application. Another focus of this big project was to produce mosses within laboratory, because collection of mosses from nature, it can be dangerous from the environment, and there are specific laws that don't allow the collection of mosses because mostly they are protected species. Concerning the standardization, just to give you an example, we use a specific moss. We employed more than 1,000 moss bags, and we studied three big areas, so Galicia, Austria, and Italy. We selected these three sites because they represent three specific environments, so continental, oceanic, and Mediterranean climate. So we wanted also evaluate the pollution in relation to the climate. We exposed the bags in several types of sites, so urban, industrial, agricultural, and background sites. We use three different types of moss bags. The rounded is the traditional one. Another one that was used several years ago, so flat style. And another one is the Mosphere. We, within the project, we realized this system, this new system. We wanted to compare the moss bags different for the shape, for the amount of moss per surface area of the bag, and also with the different diameters of the pores of the nylon net. We used the different exposure conditions, using, for example, an umbrella system or simple plastic rods for 3, 6, and 12 weeks. We wanted to see if these aspects, so the time, the height, the type of moss bag, could have some effect on the capture of pollutants. What we see, that the factors most important for the moss uptake were the amount of moss within the bag and the time of exposure. We saw, for example, that with the short period the pollution signal, so the concentration of pollutants accumulated inside the bags, was very low, but with the long period, it was a high pollution, there was a high pollution signal. So we decide, using statistical analysis, so comparing the data, we decided that the best option is to use spherical moss bags with a low amount of moss, so under 15 milligram per centimeter squared, with an elongation of 2 millimeters and an exposure period at least of 6 weeks. We apply this protocol also in other type of studies. For example, near my town, I live near Napoli, and there is a place called Land of Fires. It is famous for burning of wastes. So the contamination of the atmosphere is a very huge problem in this area. We apply, for example, the moss bags in one study within another big European project, EcoRemed, Life EcoRemed project. We studied this area, this land of fire. We selected the urban and agricultural sites, and in each site, we selected the streets and the green areas with a very short distance between the street and the green areas. For example, in this image, you can understand better. So Acerra is one city. In each city, 2 urban sites and 2 agricultural with the U and A. And in each site, so 20 meters distance, 1 green area and 1 street area. What we see that we saw that moss bags were able to discriminate not only 2 big environments, so agricultural and the urban sites, but also they could discriminate the pollution levels in areas in sites very close each other. In another study case, we went in an industrial plant, very famous, important in Europe for recycling of lead battery. And in this industrial plant, we selected several sites covering all the area. Specific, in particular, there is an area for phytoremediation. I will show you here. So within this industrial plant, we used poplar trees, Populus nigra trees, to remediate contaminated soil, because originally in these fields there were only wastes produced or stocked by the industry. So in several years we try to use plants to remove pollutants from the soil. In other cases, we use moss bags within offices and also near the industries in stocking and treatment areas of the wastes. What we saw, this is an example of evaluation we did. We analyzed the concentration of pollutants in each point. Of course, we saw that the highest values were in the treatment, in the places of treatment of wastes. The lowest levels of contaminants were within offices, so indoor environments, but also inside the phytoremediation fields. And we did also evaluation during summer and during winter to see if there were a difference between the 2 seasons. Another particular data, we analyze, of course, lead, because this industry was specific for lead battery recycling process. We saw this, there was a seasonal effect of accumulation of lead, and we saw also that the contribution, the percentage of lead can vary not only among season, but also it can be low, even if the total metal load in a specific point is high. We analyzed also moss surfaces, because in this site we use two types of mosses. This is an example of mosses used in Japan, but we used the same mosses in Italy. So sphanium has this structure like a sponge. And as you can see from the images, the yellow arrows, they indicate particles that can be entrapped not only outside the moss cells, but also within these cells. Because sphanium is a specific moss with a particular structure. If you see under microscope, it has this aspect as a sponge. So a lot of dead, empty cells that can accumulate particulate matter. In below, you see Hypnum species, another type of moss, but the surface is not like a sponge. So particles are entrapped only on one surface of these moss cells. Why is important to evaluate particulate matter in mosses? Because it is very dangerous for humans, especially if we breathe particles, especially if we breathe fine particulate matter. So smaller, very fine, very tiny particles that can arrive deeply inside the lungs. And also they are dangerous because they can transport, they can catch heavy metal or other toxic substances present in the air. So it's important that anytime we evaluate the particles, the type, the number, the size of particles that can be entrapped by mosses. For example, particles that we can observe on moss surfaces can be different in shape, in size. There you can find single particles, aggregates of particles of inorganic origin, but also biological particles, pollens, spores, as in these, or fragments of plants, as in these cases. We can analyze moss surface using electron microscope, but using backscattered electron. In this manner, you can see which are the particles more shining. So with an element, for example, lead in this case, with a high atomic number. And you can analyze chemically which type of metal is within this particle. Or you can do a mapping, so with colors or without colors, you can understand the chemical composition of the particles. In this case, for example, I just show this data from Japan, because this data I cannot publish yet, but I wanted to show you that this particle can contain also not only simple elements, simple metals, but also radioisotopes. For example, this is an autoradiography image of the moss bags, different type of moss bags exposed for different periods in the biomonitoring in Fukushima. So if you see the dark spots or the dark color, it means the presence of radioactive particles. So just to show you what I did during 2 years in Japan, we selected sites in Okayama in the south of Japan. Okayama was considered as background for comparison, so clean area to compare with the Fukushima site. So in the gray part is an area in which we cannot enter because it is still contaminated. There are still works to clean the area very near the nuclear power plant. But the other sites, there were a lot of activities to decontaminate the area. So we could go in these sites, Futaba or Kumatomioka. This is just an example because during 3 years I did other monitoring, but this was the first one I performed, in which I used the 3 most species. I analyzed several periods of exposures and several sites with different contamination levels. So I tried to standardize the MOSBAG method for radioisotopes. But other questions are related to the use of MOSes. When we use MOS, how they work, how the MOS surface can catch the pollutants. And another big question, is it still necessary to use MOSes collected from nature? Because when you collect MOS from nature, it can be dangerous because there are several laws on species protection. Sometimes it's very difficult to find one selected MOS, and they have a very high or very variable chemical composition. So the solution can be the use. So for the first question, it is necessary to study the chemical properties of MOS surfaces, because on the MOS surfaces, if you do, for example, titration analysis, acid-basic analysis, very simple analysis, you can evaluate how much charges, so functional graphs, are present on MOS surfaces. For example, carboxyl polyphenols that are important for catching ion from solutions, important also for retention of particles or other contaminants in the environment. Other type of studies is to study the MOS surface, the properties as a function of pH, of time, or metal concentration in solution, because the MOSes cannot catch pollutants in how to say, without limits. They have a limited just a moment. Sto facendo un seminario per la pasta, la macchina e la come si chiama, la Toyota, una nera, sta nel cofano. Sorry. Sorry, I was in the lab. So the system, the ability of MOS to catch contaminants is in function of pH of environment and also of the time, but also the amount of metal of contaminant in solution, because they cannot catch infinitive amount of metal. So it is necessary to study these parameters if we want to use MOSes, for example, for remediation of polluted waters. The solution, another solution to avoid the collection of MOSes in fields in nature, is to produce MOSes inside the laboratory. So within the MOS clone project, Freiburg University applied a system, a protocol to produce mosses in vitro. So using different media, solid or liquid media, and also bioreactors, so 5 liters or 12 liters of production. And specifically, we studied Sphanium palustre. Sphanium in general is an important moss because it is very important for the formation of peatlands. I don't know if you ever heard about peat and peatlands, la torba, I don't know in Spanish, but peatlands are important sink of carbon. So in the carbon cycling, they are very important for our planet because they can store a huge amount of carbon. So climate changes are very dangerous for peatlands because with the high temperature, the sink function of peatlands can be damaged. So instead of capture carbon, they can release carbon, so increasing the problem related to presence of CO2 in the air, in the environment. But also peatlands are important because they are considered historical archives of atmospheric deposition of several years ago. So if you study the profile of peatlands, you can understand which was the atmosphere of several years ago, just studying the peatland profile. So this moss, I think, is more important between the other type of moss. We selected this moss and we try to characterize not only the morphology, the genetics, but also the characteristic of surface and the ability of moss to catch contaminants, not only in vitro experiments, but also using moss bags. For example, we compared with the field moss, so the same moss species collected in nature, and we did different analysis. And also we compared with moss bags applications with another type of moss. And in general, we saw that mosses produced in the lab is a very ready to use material. We can produce in lab and use directly, avoiding treatment in collection in nature. So we avoid also the produce damages to the environment. And also we saw that it is effective in the adsorption of contaminants, and especially it has a very low and uniform baseline element content that is important if, for example, we want to monitor the pollution levels in background or clean environments. So that is all. I finished with my presentation just to give you an example of what we do with mosses. I hope that it was useful for you. So if you have questions, please tell me. This is me in Japan searching for moss. But if you have a question for everything, I don't know if you have time. Sorry if I employed a lot of time to explain.
Anna, I have a little curiosity.
Sí.
Is it possible, or maybe mosses can play a role?
Okay. Sorry.
No, sorry. Particulate matter is
Sí.
important for diesel engines in cities. Does mosses have any role there? I mean, is it possible to use mosses to reduce the diesel contaminants, particulate matter especially?
You mean as filters?
like to spread mosses to reduce the contaminants.
To reduce. There is a UK research group. They realized some city installations, some like big panels with mosses to use as biofilters of pollutants. I think this can be a good way to sensitize people, so to let people to understand that mosses can be used for catching pollutants. But I think that the biggest problem is they do this type of filters, this type of panels for reducing pollutants without studying the moss properties, because they have limits. For example, if you use living moss, they can be managed damaged by the weather. They can how to say they can die during time, so their efficiency can decrease during time. And if you don't know which is the maximum, which is the maximum amount that maximum amount of pollutants that mosses can catch, I think it's difficult to use this type of system without a very specific scientific knowledge of the use. Someone used the mosses for remediation of polluted waters, but also in this case, you need to study the chemistry of surfaces. So this type of experiment, at least to understand how much is the maximum amount for a specific moss. If you do this type of studies within the lab, after that you can apply for remediation studies. But I think that nobody do that. Only few people, sometimes very few data in literature concerning the ability of moss surfaces. So they apply without knowing the moss properties, the chemistry of moss. So this topic, research topic, I think it's important and should be researched, should be studied more to understand if it is really impossible. Is really possible to use mosses for remediation pollutants.
Thank you very much. I agree. It's required.
I hope I was clear.
No, it was very
this aspect is very complex, but I prefer to use mosses as a biosensor, as indication of. So catching pollutants and analyze the type of pollutants. But if you want to use for remediation, for cleaning water or air, I think that we need to study more and more. And me and a few other people are doing this type of studies. So the chemistry of moss surfaces.
Thank you very much. Any other question? No.
I know the topic is very huge, so I tried to give some input for further discussion. So
it was very interesting, Anna.
Yes, a lot of things to do, and to resume is very difficult. But anytime I can explain everything.
Thank you very much. See you. Bye.
Bye.
Creo que podéis descansar 5 minutillos y vemos el segundo. Luis? No, I'm going to call Luis in case he wants.
Okay.
I am here again.
Okay.
Thank you very much, Anna. Can
you.
ask to Cristina?
Here,
Cristina, please stop recording.
Right.
Anna, thank you very much. We keep in touch because
Okay.
for two reasons. Because I had to send you the certificate of this year.
Okay.
And I don't know, and we conspirate to looking for any interaction with your team, with your lab.
Yes.
Now it's time to rest.
Yes.
But we keep in touch.
Okay.
Cristina, I am happy you meet Cristina because she's a good colleague, and she works in remote sensing, and she's very close to other approach about the environmental control, pollution, etc., but using remote sensing.
Good.
So in the case we look for interaction or any kind of project,
Yes.
probably we keep in connection with Cristina. I am sorry because I want to hear you, but I am working with you know, the hammer and the tool with the operari. So
Sí.
the students more or less follow you, the students.
Sorry?
The students have been following you. More or less. Okay. So
but in any case, I can explain. So
okay.
Don't worry. Also,
I have to come back to the lab with the operari.
Okay. See you. Ciao, Luis. Ciao.
Ciao. Thank you very much.
Gracias por todos.
Gracias. Ciao.
Ciao.
Creo que